Everyone, my name is Avin Vahdaninia and today I will be presenting my project on the expression of TP53 in pEGFP-N1, a fluorescent localization approach. This project focuses on cloning TP53 cDNA into a mammalian expression vector to study its localization in cells. So, the aim of this project is to clone TP53 into pEGFP-N1, a mammalian expression vector, and validate its expression in cells. This allows us to track TP53 localization via GFP fluorescence. Why TP53? I choose this because TP53 is a crucial tumor suppressor gene that regulates a cell cycle progression, apoptosis, and DNA repair. Mutations in TP53 occur in more than 50% of human cancers, leading to uncontrolled cell proliferation. Understanding its localization and expression patterns can provide insights into its function in disease contexts. So, to study TP53 localization, I selected pEGFP-N1, a mammalian expression vector with key advantages. And one of them is CMV promoter, which ensures a strong gene expression in mammalian cell. And also EGFP tag allows a direct visualization of TP53 localization. MCS, or multiple cloning sites, before GFP ensure in-frame fusion with TP53. Neomycin resistance allows selection of stable transfectants. This vector is well suited for our goal of tracking TP53 localization in living cells. To clone TP53, I designed forward and reverse primers. Flanking the gene, these primers contain X-hole and EQR1 restriction sites for ligation into pEGFP-N1, a ribosome binding site or RBS to ensure proper translation. A sequencing primer in the CMV promoter to confirm TP53 integration, and also by amplifying a TP53 cDNA with these primers, we prepare it for cloning into the vector. As you can see, I have the full cDNA sequence here, and as you can see, we can see the forward primer and also reverse primer here. So, the cloning process here follows these steps. PCR amplification of TP53 cDNA using specific primers. Restriction enzyme and digestion of both the PCR conduct and pEGFP-N1 using X-hole and also EQR1. Ligation of TP53 into the MCS of the vector, ensuring in-frame fusion with EGFP, and transformation into E. coli to generate recombinant plasmids, validation via colony PCR, restriction mapping, and sequencing before transfecting mammalian cells. This workflow here ensures proper TP53 cloning and expression. Here, in this workflow, which is really complete, you can see the template in the PCR amplification, the buffer in the restriction digestion, and the ligase in the ligation, and everything here is really obvious. So here is the final plasmid map showing key components. CMV promoter drives TP53 GFP expression. MCS allows cloning of TP53. EGFP tag enables fluorescence tracking. Neomycin resistance provides selection for stable transfectants. This confirms that TP53 is correctly inserted and also expressed. So, as an alternative, I considered another vector with CMV promoter for high expression, BAM-H1 X-hole MCS as an alternative cloning site. HIS tag for protein purification instead of fluorescence tracking. Neomycin resistance for selection. This alternative would be better suited for biochemical studies rather than localization experiments. So, to ensure TP53 is correctly inserted, I will perform a cloning PCR to confirm the presence of TP53 in bacterial colonies, restriction digest analysis to verify correct orientation, Sanger sequencing using a CMV primer to confirm the TP53 sequence. And these steps ensure the plasmid is properly constructed before moving to mammalian expression. To validate TP53 expression, I transfect HEC293 cells using a liposuction or electroporation use fluorescence microscopy to confirm TP53 localization and perform Western blot analysis to detect the TP53 GFP fusion protein. These experiments will confirm successful TP53 expression and localization in living cells. In summary, TP53 cDNA was cloned into pEGFP-N1 for localization studies. Cloning was validated through PCR restriction digestion and also sequencing. Functional expression will be confirmed using fluorescence microscopy and Western blot. Understanding TP53 localization can provide insights into its function in stress response and also apoptosis. The methods and reagents used in this project were based on protocols from SnapGene, NEB, and other molecular biology resources. And thank you for your attention, and I would be happy to answer any question in the comments section.